my name is Peradonna.
Hi, my name is Lavinia Esther.
Hi, my name is Ariana.
Hi, my name is Elvic Carlson, and we are from Group 9.
And lastly, experiment 3, physical properties of different lipids and fatty acids.
The objective of this experiment is to determine the use of the DNS method for quantifying sugar's concentration in given sample. The apparatus and materials needed in this experiment include microcentrifuge tubes, spatula, dropper bottles, distilled water, toluene, olive oil, shortening, and G.
2 sets of 3 separate microcentrifuge tubes were prepared. 5 drops or a small amount of solid, 3 to 4 granules of all the lipid-containing products that were brought were added. Their appearance was described in the table. Their smell was also described in the table. For set 1, solubility in polar solvent, about 1.0 milliliter of water was added to each centrifuge tube. The tubes were capped and shaken to mix. The observations were recorded in the table. For set 2, solubility in a non-polar solvent, 0.5 milliliter toluene was added to each sample. The tubes were capped and shaken to mix. The observations were recorded in the table.
This is the results for physical properties of different lipids and fatty acids. The observation of the appearance are olive oil is clear, pale yellow color and in liquid state. For shortening, it is white color and semi-solid state. For G, it is yellowish color and in semi-solid state. The observation for the author are olive oil is mild and have characteristic oily smell. Shortening have faint fatty odor and G have strong buttery aroma. For their solubility in water, olive oil are insoluble and form separate oily layer. Shortening are insoluble and remains as solid or fat globules. And for G, it is insoluble and floats as oily layer. For their solubility in toluene, olive oil are soluble and forms clear homogeneous solution. Shortening are soluble and dissolve, forms cloudy to clear solution. G are soluble and dissolve well, forms uniform mixture.
In conclusion, this experiment successfully investigated the physical properties of different lipids and fatty acids based on their appearance, odor, and solubility in polar and non-polar solvent. The results show that all lipid samples were insoluble in water but soluble in toluene, which indicated their hydrophobic nature. This observation confirmed that lipids share common physical properties, especially their non-polarity and low affinity for water, which explains their classifications as lipids. This practical improved the understanding of lipid characteristics and their presence in everyday food products, as well as the relationship between chemical structure and solubility behavior.
We learned that lipids are classified together based on their shared solubility properties. We face challenges handling hazardous chemicals like toluene, which required strict adherence to safety protocols. And we suggest that the experiment could be improved by testing a wider variety of lipids, such as comparing saturated and trans fats.